Transcription is the first step of gene expression where the enzyme RNA polymerase copies DNA to mRNA.
There are 3 stages of transcription: initiation, elongation, and termination.
The DNA strand has a promoter and a terminator region.
The area in between these 2 points is known as the transcription unit, which is where transcription occurs.
To the right of the promoter is known as the downstream, and to the left of the promoter is the upstream.
In the upstream is the Tata box, which helps with the positioning of the RNA polymerase.
The process of transcription begins with initiation, when an enzyme called RNA polymerase attaches to the DNA upstream of the promoter and begins the process.
This area contains information to specify the amino acid sequence.
The transcription factors bind to the RNA polymerase and the promoter and separates the 2 strands.
One strand becomes the template strand and the other one becomes the non-template strand. The mRNA is created based off of the template strand.
The second step of transcription is elongation. RNA polymerase moves downstream along the DNA strand and along the way synthesizes the mRNA.
RNA polymerase generates from 5' to 3'. When the RNA polymerase hits the end of the gene, the next step begins.
The last step is termination. During this step, mRNA processing and RNA splicing occurs.
First, RNA polymerase unbinds from the DNA strand. RNA polymerase II then transcribes the sequence which codes for polyadenylation signal, which is known as AAU-AAA. Proteins then cut the pre-mRNA free from the polymerase. The pre-mRNA strand is now processed. The enzymes in the nucleus modify the pre-mRNA before it can release into the cytoplasm.
During the modification of the mRNA strand, a methyl cap is added to the 5' end and a poly A tail is added to the 3' end.
The cap and the tail have 3 main functions: to protect the mRNA from damage, to help the mRNA leave the nucleus, and to allow ribosomes to attach to the mRNA when they get to the cytoplasm.
The RNA splicing is basically a cut and paste process.
The splicing occurs through the spliceosomes, which are multiple small nuclear ribonucleoproteins that are found in the nucleus of the cell and are made up of RNA and protein molecules.
Spliceosomes have the ability to recognize the splicing sites.
RNA splicing cuts out sections of the RNA molecule, which are called introns and exons.
Introns are non-coding segments that are located between coding regions and are the ones that get removed.
Exons are the remaining regions that eventually translate into amino acid sequences. Once the introns are removed, the exons are spliced together.
The finalized strand is then ready to leave the nucleus.